Welcome to this lecture on cell and molecular biology. Today, we're diving into one of the most fascinating topics in biology, the genetic code. Imagine having a secret message that holds the instructions for building every part of your body. How does a cell read this message? We will explore how the simple four-letter language of DNA is translated into the complex proteins that keep us alive. Let us begin our journey into the language of life. To start, let us think about why we even need a genetic code. Your DNA is like a massive instruction manual written in a very simple language of only 4 chemical letters. However, the actual machines that do the work in your body, called proteins, are built using a completely different language, made of 20 different building blocks, called amino acids. The genetic code is simply the biological rulebook, or dictionary, that your cells use to translate the 4-letter language of DNA into the 20-letter language of proteins. It bridges the gap between your genes and your physical traits. Now that we understand what the genetic code does intuitively, let us look at its formal definition. Formally, the genetic code is the set of rules by which information encoded in messenger RNA is translated into proteins by living cells. The fundamental unit of this code is called a codon. A codon is a specific sequence of exactly 3 consecutive nucleotides. Because the cellular machinery reads the sequence one triplet at a time without sharing bases between adjacent codons, we call it a non-overlapping triplet code. This precise reading mechanism ensures that the protein is built correctly from start to finish. Building on the idea of codons, there are 2 incredible properties of this code we must highlight. First, it is almost entirely universal. Every living organism, from bacteria to blue whales, uses the exact same dictionary. This universality is the foundation of modern biotechnology, allowing us to put a human insulin gene into bacteria to produce life-saving medicine. Second, the code is degenerate, which means it is redundant. While one codon specifies only one amino acid, a single amino acid can be specified by multiple different codons. This redundancy acts as a crucial buffer against harmful genetic mutations. This brings us to an important question. Why does the genetic code use exactly 3 letters per word? We can understand this by looking at the simple mathematics of combinations. The formula C equals b to the power of n helps us calculate the number of possible words. Here, C represents the total number of possible codons, or combinations we can make. The variable b represents the number of different bases available in our genetic alphabet, which is exactly 4 for RNA: adenine, uracil, cytosine, and guanine. Finally, the variable n represents the length of the codon, meaning how many bases are grouped together to form a single word. If we increase the length of the codon, the number of possible combinations grows exponentially. Now that we have our formula, let us test it to see why a 1- or 2-letter code fails. We know we need to code for a total of 20 different amino acids. If the cell read the code 1 base at a time, setting n equal to 1 would give us 4 to the power of 1, which is just 4 combinations. This is clearly not enough to cover 20 amino acids. What if the cell read the code 2 bases at a time? Setting n equal to 2 gives us 4 squared, resulting in 16 combinations. While closer, 16 is still less than 20. Therefore, a doublet code cannot possibly specify all the necessary building blocks for proteins. Since a 2-letter code is insufficient, let us apply our formula to a 3-letter code. By setting n equal to 3, we calculate 4 to the power of 3. This means multiplying 4 by 4 by 4, which gives us 64 possible combinations. This number is more than enough to cover all 20 amino acids, which perfectly explains why the genetic code must be a triplet code. The extra 44 codons are not wasted. As we discussed earlier, they provide the redundancy that protects against mutations, and a few of them act as special stop signals to tell the cell when to end protein synthesis. Now that we have proven mathematically why the code uses triplets, let us look at how the cell actually reads a genetic message. When a messenger RNA sequence is ready, the cell's ribosome acts as the translator. It takes the continuous string of letters and breaks it down into non-overlapping triplets, starting from the very first letter. This establishes what we call the reading frame. Once the sequence is divided into these 3-letter blocks, the ribosome uses the standard genetic code dictionary to translate each codon into its corresponding amino acid, linking them together to build a protein. Before we move on, let us see this translation process with a concrete example. Suppose we have a short messenger RNA sequence that reads AUG CCG UAA. Our goal is to determine the resulting amino acid chain. First, we divide the continuous sequence into distinct blocks of 3 letters, AUG, then CCG, and finally UAA. Next, we translate each block. The first codon, AUG, is the start codon and codes for methionine. The second codon, CCG, codes for proline. The third codon, UAA, is a stop codon, which tells the machinery to finish building. The final physical result is a tiny protein chain consisting of methionine attached to proline. To make this process completely clear, let us look at a diagram of what we just calculated. In the center, you can see the horizontal messenger RNA strand divided into our 3 codons, AUG, CCG, and UAA. Below the strand, transfer RNA molecules act as the physical adapters. The first adapter binds to AUG and brings the green methionine sphere. The second adapter binds to CCG and brings the blue proline sphere, linking it to the methionine. Finally, look at the UAA codon on the right. Instead of an amino acid, it triggers a red stop signal, terminating the process. This diagram perfectly illustrates the step-by-step assembly of a protein. Seeing how precisely the codons align in our diagram brings us to a critical vulnerability in the system. Because the genetic code is read in continuous, non-overlapping triplets, the exact starting position is absolutely crucial. This grouping is called the reading frame. If a mutation occurs where a single base is inserted or deleted, it pushes all the subsequent letters out of alignment. This phenomenon is known as a frame shift mutation. Unlike a simple substitution that might change only one amino acid, a frame shift mutation alters every single codon downstream of the error, usually destroying the protein's function entirely. Let us demonstrate the devastating effect of a frame shift mutation using our previous sequence. Our original sequence produced methionine and proline. Now, imagine a mutation inserts a single extra uracil base right after the very first letter. The new sequence becomes AUU, GCC, GUAA. When we apply our strategy of breaking this into non-overlapping triplets, the reading frame shifts. The first codon is now AUU, the second is GCC, and the third is GUA, leaving a single A hanging at the end. Translating this new set gives us isoleucine, alanine, and valine. A single added letter completely destroyed the original message, creating a totally different non-functional protein. We have covered a lot of ground today, so let us summarize the most important ideas. First, the genetic code is the universal dictionary that translates the nucleotide language of DNA and RNA into the amino acid language of proteins. Second, this code is built on non-overlapping triplets called codons. Because there are 64 possible combinations for only 20 amino acids, the code is highly redundant, which helps protect ourselves from certain mutations. Finally, we saw how sensitive this triplet reading frame is to insertions or deletions. Understanding this code is the absolute foundation for studying how life functions at a molecular level. Great job following along.